Hello?
Right. And then a very
Hello?
Sir, any issue?
Lowry method. It was very highly cited paper. In Lowry method, actually, I've written here number 1 Lowry method. A is the Lowry method.
Hello?
Sir is talking from Google Meet, sir.
Hello, Dr. Khan?
Sir, he's talking from Google Meet.
Which absorbs the 7.
Yeah, but it's not audible here.
Not audible?
They all
Yeah,
absorb,
nothing is audible.
nanometer wavelength. So Lowry method gives blue color
Here the sound is very clear.
and blue color mostly below above 700. So maximize absorption, maximize.
मैडम जी पानी दीजिए ना प्लीज वो सामने इसमें नहीं है आपको दिखाऊं पास आज दोनों को स्कूल नहीं भेजे
Hello? Yeah.
No, Tenzel,
Post-translational modification. As mentioned in the beginning only, that there are large number of modifications. Almost 400 different modifications of proteins are known. Multiple modifications, combination of modifications, and some I have given example here. Some examples, although there are hundreds, but phosphorylation, lipidation, ADP ribosylation, cleavage of protein, ubiquitination. If I talk some of this, I will show you, especially phosphorylation and lipidation and cleavage. But I'm sure all of you know ubiquitination is very good. This is important that once protein is marked with ubiquitin, it is destined to destroy by proteasome. Our cells have got machinery called proteasome, and those proteasome can degrade those proteins. This is a marking. Once it is ubiquitinated, it can be cleaved. It can be degraded by proteasome. Glycosylation, many proteins are glycosylated, and those glycosylated proteins actually are on the surface and many places. Hydroxylation, many of our proteins in the bones are hydroxylated. Sulfation, sumylation, I'm sure you know gene expression. There are proteins, actually histone proteins, which are stylated and methylated, and they regulate the gene expression. So I have mentioned only few, but there are hundreds. Now, you can imagine that if a protein gets multiple phosphorylation, multiple lipidation, multiple ubiquitination, combination of phosphorylation and lipidation, combination of these modifications. So that is going to create large diversity. So I will say that protein modification increases the diversity into the protein, and that allows the protein to regulate multiple process, activity, and interactions. We have been working on many modifications. Phosphorylation is a very common, which I will show you today. What is the role of the phosphorylation? I have written here one example also. Phosphorylation of a transcription factor increases affinity for promoter, or it can even decrease the affinity for a promoter, very commonly. So let's see why needed. So imagine that while you are sleeping, sometimes you are running, sometimes you are not eating, sometimes you are eating more, sometimes you need more energy. It is winter time, so more for eating, your body needs more energy. So this time point, different rate of metabolic pathways. Now, if you think that how you will control the different rate of the metabolic pathways by enzymes, either you have for each rate of the, you have to have different enzymes, which will be probably different in sequences. So you can imagine, then you will require millions of genes in your genome, and then those many proteins will be very difficult to control in their interaction, their regulation. But if you have the ability that you can phosphorylate a protein, and that is going to change its structure a little bit, but it is going to modulate the activity of the protein, then it is great. You add one phosphate, the activity will go up by 2-fold.
Madam, join Gammani leave.
I'm trying to say that these modifications, whether it is a phosphate or any other modifications, can change the local structure of the protein. Let me 1 minute discuss about it. As I said, when we are talking about the protein structure, that one sequence will give always one structure. And that is because the bond, whether it is a ionic bond, whether it is a hydrogen bond, disulfide bond, whatever it is, are fixed based on the sequence of the protein, amino acid of the protein. But once you add something additional, or if a ligand bind to the protein, it is going to bring different kind of charge or hydrophobicity. Now, if the charge changes into the protein, then the folding is going to be again changed, and it will be different than the original when it was not modified or not phosphorylated. So, point I want to make, that phosphorylation in a protein, which happens on a certain amino acid, for example, serine, threonine, tyrosine, histidine, aspartate. So these addition, because it has phosphate has got a negative charge, the structure is going to change locally, and that change into the structure is going to allow the protein either to increase the activity or decrease the activity, or increase or decrease its interaction. For example, if it is enzyme, the activity may increase or decrease. If it is a transcription factor, it may bind to DNA with more affinity or less affinity. So it is so easy now. Instead of making millions of protein or genes, incorporating genes into genome, nature designed that modifications can fine regulate the metabolic pathway, the rate of the metabolic pathway. What types? Some I showed you, and who does it? It is many times it is done by enzymes. So let's see some example of this. First example I have given metabolic pathway regulation. I'm sure you all read in MSc classes, you all read glycolysis, where sugar is broken down into 3 carbon molecule pyruvate, which goes to the mitochondria, where enzyme converts to acetyl-CoA, enters into the TCA, NADH, all those things are generated. Now that enzyme is a complex enzyme. Pyruvate dehydrogenase is complex. And that regulation comes from the phosphorylation. When it is phosphorylated, then the enzyme becomes inactive. So you can imagine that instead of creating different activity having enzymes, nature created that if you phosphorylate, so if there are thousands of units in that enzyme, please try to understand, suppose there are 1,000 polypeptides in that complex and they are all working, suppose 5 gets phosphorylated, the activity will reduce little bit. 100 phosphorylated, reduce much more. All phosphorylated, completely it will stop. So that is the way the gradation in the activity was generated by just the phosphorylating the enzyme. Similarly, I'm sure you have read about glycogen phosphorylase, glycogen synthase. They all are regulated by phosphorylation. HMP pathway by phosphorylation, fatty acid synthesis, you name it. Many pathways are actually regulated. Metabolic pathway in our cells are regulated by phosphorylation. Another modifications, I said I'll tell you some of them. Another modification is lipidation of proteins. Lipidation means addition of the lipid to the proteins. And this modification is always covalent modification. This is a covalent attachment of lipids to the proteins. And then they can regulate multiple functions. For example, if a lipid is attached to the protein, then it can go and insert into the membrane because membranes are hydrophobic. Signaling pathway can be regulated. Trafficking can be regulated. When the vesicle trafficking happens, many of the RAP proteins actually help in the trafficking of the vesicle to the different organelles, delivering the proteins. So I'm trying to say that lipidation of the protein also helps in various ways, which is signaling, trafficking, membrane anchoring, and all those things. G protein, for example. So G protein needs an attachment, but although it has to go out, it has to go eventually into the cytosol. So there should be such attachment that it can come out. Now, if I tell you what kind of modification happened, you can see here, I have mentioned that there are milstrilation, prenylation, so many cholesterylation, GPI enter, phosphatidylmethylamine addition, palmitic acid addition, but many different kind of lipids can get attached to the protein for various kinds of functions. I'm showing you a very great example. For example, if you have to turn on an electricity light in your room, you just switch on off. This lipidation or addition of a fatty acid in the protein works like that, on and off. So if you see the green colored protein and a purple colored fatty acid here embedded into the membrane, now see a situation if a ligand binds or a phosphorylation happens, the charge is going to change, the protein structure is going to change, and this is going to change in such a way that it shrinks the protein, which was in a larger volume, in a smaller way, because the structure is changing. So it will pull the fatty acid, and that is how the protein can get released from the membrane. So molecular switches just by modification by a ligand. Of course, there are many different kind of switches. I'm not going to enter into that, but you can read it. It's a very elegant mechanism in the cells. It's called protein myristoyl electrostatic switches. This plus, actually, somebody might be wondering what is this plus. These are actually basic amino acids. Since membranes are negatively charged towards the cytosol, this positively charged amino acid help in stronger binding. So already the fatty acid was in the membrane. The positive charge also helped in a little stronger association. But when the structure changes by a ligand or by a modification, then the structure pulls the fatty acid, and now it is free to go into the cytosol. I'm going to show another modification. I'm sure all of you know this modification very well. Many of our proteins which are secreted, for example, digestive enzyme, they are produced, synthesized as a zymogen or inactive enzyme, pepsinogen, thymotrypsinogen. So I'm trying to say that they are synthesized when they are not active. The reason is that their active site is blocked. So therefore, substrate cannot reach to the active site. But nature has designed such beautifully that once it's, for example, this pepsinogen will seize the low pH, it gains autocatalytic activity, autoprotease activity, and it cleaves a small peptide. As a result, this blue part of the protein goes away, the active site gets open, and the pepsin becomes inactive. Similarly, the trypsin and chymotrypsins are also activated, which help in digestion of the protein. Many blood coagulation cascade, all these things you see that there are a small cleavage, and those cleavage are going to make the protein active. So removal is because the structure is going to change because you are removing some amino acid, and I have been repeating that a sequence of a protein always gives a certain definite structure. In this case, when you remove the amino acid, the structure is going to change, and in this case, the active site opens, and now it can do the job. I'm not giving you many examples, but it's still some. This is a rare amino acid again, present in the elastin, and this was created by modifications. There are 4 lysines, so sometimes you see your cartilage is very elastic. That is why your knee, you can bend the knee. If there were 2 bones, bending would not have been possible, and those are the joints. Actually, elastin are required. Connective tissue, your blood vessels, they are not supposed to be very, or should I say, rigid. They are supposed to. They should have elasticity. So these elasticity is provided by glycosylation. How? Because this 4 lysines join together to make this molecule, which actually it has been tested. I have written here. It has been tested to show that pyridinium core of desmosine remains intact even at very high collision energies. So this is a modification of a protein where the rare amino acid comes and joins the protein. There are many such modifications. I'm not going to show you all of them, but as I said, there are hundreds of modifications which actually help so that we can perform various functions. But I'm going to do focus about phosphorylation more. One of the reasons is that phosphorylation is the most common modifications. There are kinases, and those kinases actually are going to add a phosphate group on the protein, and then a phosphatase will remove the phosphate group and protein can come to the ground state. By doing this, in the beginning also I mentioned that by adding a phosphate group which has negative charge, the small structure changes allow the protein to change the structure, and their interaction, their activity can change, and that is how they regulate multiple processes, metabolic pathways, cell division, growth, signaling, and even they can regulate virulence, or they can regulate survival required for survival in bacteria. Amazing that human has got more than 500 kinases and 200 phosphatases, and that shows that how important these kinases and phosphatases are, because they run the show. They regulate your energy production. They regulate your cell division. They regulate your growth. They tell the cells when what to do. So signaling. So that is why phosphorylation is a very important modifications for the proteins. What happens in this? The kinases take ATP. They bind to the ATP. They always got a ATP binding domain. So they will bind the ATP, hydrolyze the ATP, and take the phosphate and add it on the protein. Once the phosphate is added on the protein, because of the negative charge on phosphate, the structure is going to rearrange, and some local structural changes will happen, and that is how the phosphate is going to regulate the activity or interaction of the protein. Now, once the job is done, when the cell wants now the enzyme or protein should come to the basal level, then again phosphate can be removed and there are other enzymes. So these kinases and phosphatase themselves are also regulated by signaling pathway, and they can do they can modulate all kind of reactions in our metabolic pathways, growth, and everything which I have been mentioning. Surprisingly, what we and many other people also noticed, that these modifications are also work in bacteria as a virulence factor. So bacteria has seen that how do our pathways work. So they have utilized this knowledge in evolution, evolving such proteins which can modulate our metabolic pathway, and I will show you that. So talking about bacterial proteins, especially in phosphorylation and involved in signaling, there are two sets of major enzymes. One set of the enzyme is called two-component system. In two-component system, as the name implies, there is one component which is histidine kinase, and the second component is called response regulator. This histidine kinase is present into the membrane, so it is exposed outside. And once it is exposed to outside, this can sense what is happening outside in bacteria. So when bacteria somewhere is growing, these enzymes, this kinase, histidine kinase, can always sense what is the temperature outside, what is the pH outside, food is available or not, many things they can sense. And that sensing actually passes signal to the inner response regulator, which actually based on the signal eventually gives a response. So when a signal comes, gets phosphorylated, because of phosphorylation, again the structure will change, which will allow the histidine kinase to interact with the response regulator. It will transfer the phosphate to this, the structure of response regulator will change. And now this is a kind of a transcription factor. So it was not interacting with the DNA before, but once it is phosphorylated, it can bind to many, many promoters and make those proteins which are now required to cope or to deal with the signal which has come from outside through histidine kinase. Once the signal allows expression of the genes, new proteins, and they can now help the bacteria or microbes to survive in the new environment according to the signal. The other set is called serine, threonine, tyrosine kinases, and
I have completed mine.
those kinases, many of them are also on the membrane. They can also sense outside, but normally they do not. Some of them do bind to the DNA, but many of them actually, once they are activated, they actually phosphorylate a substrate. All our metabolic pathway are regulated by serine, threonine, tyrosine kinases. And once for example, some enzyme is phosphorylated, so then the activity of that enzyme may change. Either it becomes high or become less, and then so what you can say that serine, threonine, tyrosine kinases are able to modulate the activity of the protein. And as I have been mentioning many times, that they can regulate cell division, metabolism, phosphorylation in bacteria, and even that's what I'll show you, because you can read about all these things, but virulence and survival I have been working also, and it is a relatively recent thing that how they regulate the virulence and survival of the bacteria. This is a macrophage. I'm going to tell you about the importance in immunology, your innate immunity, how the phosphorylation is important for our innate immunity. So this is a macrophage, and I'm sure all of you know that these macrophages, resident macrophages they are in our lung, in our tissues, monocytes are in our blood, and their job is that when a particle or a foreign particle or a pathogen or a bacteria or foreign particle comes, they will engulf it and then they will take it to the lysosome for degradation. Now, once they take it for degradation and take it to the lysosome, this route is actually fixed. It is not that this vesicle is left. Eventually, it goes to the lysosome, but this route of traveling is actually guided with the help of a modifications and proteins. There are phosphorylation which help in acidification, acquirement of the acidic pump by the lysosome. So if the phosphorylation does not happen, then the lysosome will not be acidified and enzymes will not work, and whatever comes may not get degraded. That was, maybe I'll try to explain this in another way also, that when something is phagocytosed by macrophages, they reach to the destiny, their lysosome, and this is a fixed pathway guided by proteins and modifications. It is something like, suppose you are mailing an envelope. When you mail a letter, then you write name of the person, colony name, city name, town, all those things, state. Imagine that if one of the line is getting deleted, either it is colony name or city name gets deleted somehow, then your letter is not going to reach to whom you have mailed. That's what happens in this mechanism. That if some protein gets messed up, which actually guides the vesicle to the lysosome, or if some modification is messed up, then actually the job will be not done by the macrophages. And this knowledge was actually learned by the bacteria. Bacteria learned that how our innate immune system or phagocytosis and macrophages, how do they work. So eventually they degrade, but if modifications happen, they will not be able to degrade. So talking about the bacteria, human, I have already told you there are 500 kinases. Bacteria, for example, this bacteria has got 11 kinases, 1 phosphatase, 2 tyrosine phosphatase, and 2 tyrosine kinase. Many of these kinases perform various functions. It also has 2 tyrosine phosphatases. And what we've noticed that if I knock out this tyrosine phosphatase from the bacteria and I grow this bacteria, nothing happened to the bacteria. It was able to grow fine. There was no problem. But when I used this knocked out strain for infection, then we found that it was too locked down. That means the infectivity was reduced. So that showed that these phosphatases were not meant for bacterial survival or something like that, but rather it was meant that it can modify our protein so that it can survive them. So when Mycobacteria or the bacteria which causes tuberculosis, when it is phagocytosed, they will secrete this tyrosine phosphatase. It will modify the pathway, so it will not end up into the lysosome, or the lysosome will not get acidified, so the bacteria will be able to survive. So you can imagine the power of the phosphorylation and modification, that how our innate system uses, and how these days, or now we understand it better, that what are the ways they manage to survive in our cells. Similarly, I'm not going to the detail, but we noticed that one phosphatase was also very important for survival of this is a serine threonine phosphatase. If I knock out, it was essential gene, so bacteria was not able to survive. So we complemented and then grew the bacteria. So there was multi-septa formation, all those things. I will not go into the detail, except that I would like to mention that this enzyme was important for survival of the pathogen. Similarly, we have been working on another bacteria, and I will data about this, but we have been working on many kinases and phosphatases. And knocking, you see here, it makes very long chain, green color. On the right side down, you see green color chains. It makes chain-like structure, very long chains. And we noticed that these long chains are made, they are regulated by a kinase. So pathogenesis of the Bacillus anthracis or the anthrax-causing bacteria is also through the phosphorylation and dephosphorylation. As I said, I'm not going to tell you the detail, but you can see here, there was a wild-type bacteria, very long chain. Then we knocked out the gene, this kinase, gene P, protein kinase C, and you can see short chain. We complemented, again, we got the long chain. So I'm trying to just say that these kinases and phosphatases or phosphorylation plays very important role in regulating multiple functions in the cell. Similarly, many other modifications of the proteins allow cells to regulate multiple pathways. That is how everything happens into the cell. With this, I will end the already, I think, about 1 hour or so. So I will take your questions now. Thank you very much. Can you hear me?
Yeah, sir, you're audible. But actually, the participants are in Zoom and we are in Google Meet actually.
Oh, okay. So if anybody has any question, I'll be happy to answer. They will not be able to tell me question, isn't it? Was it okay? The slides were okay?
Yeah, slides were okay actually, but I kept some time mute myself in Zoom, so it was a little bit issue for some time. I have kept it done.
Okay, all right. So is there any question then?
Right now,
if somebody is writing any question.
I don't see any question right now.
All right, if there is no question, then thank you very much, Dr. Khan. Sorry, I think I should have presented only in the Zoom only. I don't know why it did not work this time.
Yeah.
Maybe something post-hearing needs to be done, but anyway.
Yeah,
so
sir.
Thank you very much. Dr. Muralidhar also may not be able to hear me.
No, afterwards he was able to hear to you.
Okay. All right. So thank you. Can I leave now, or you want me to stay past?
Yeah, I will stop Google Meet now. We will go directly to Zoom now.
Dr. Yogendra Singh.
Yes, sir.
Thank you very much for a great lecture. Unfortunately, first half an hour I could not hear anything.
Yeah, technically it was my ignorance. Actually, I was thinking when to say YouTube like Zoom will have if I myself kept mute, it will be working like that. So it was little bit my ignorance.
That's okay. That's okay. I just wanted to thank Yogendra Singh for the time and the lecture that he has given. Probably we should get him again for more detailed and more clear things. Okay.
All right. Thank you very much.
Thank you, sir.
Yeah.
Thank you very much, sir, for accepting the invitation and giving us the address. Sir, now it is your lecture. You can start sharing the screen and start the presentation. Sir, are you there?
Yeah, I'm there, but it says host disabled participant screen sharing.
Yeah, I have started now. It's a default setup actually. I have given the share and now it is coming. Yeah.
Can you see?
Yes, sir.
Okay. Am I audible?
Yes, you are clear.
I didn't see any response from students in the previous lecture. I hope there is audience there.
Yeah, they are there. Our campus students are a little bit mute, but other outsiders are making their camp. Now and then they are unmuting themselves by default.
Noise is not the point. I would like the students and junior faculty to respond to the speaker by asking questions. Otherwise, we don't know whether anybody is understanding anything. It will go waste, isn't it?
Yeah.
Anyway, once again,
sir, make it slideshow, sir. Make it slideshow.
Yeah, I'll do that. Is it okay?
Yes.
All right. Once again, friends, you have heard already 3 lectures. This is the second lecture that I wish to discuss with you, which is linked to the previous lectures. This is on immobilized enzymes and antibodies, preparation and application. As I mentioned, this is a thematic workshop, lecture workshop on some aspects of proteins. We may not be able to cover everything, but at least conceptually we have gone through. You have heard the basic aspects of protein structure. And one of the things that Dr. Yogendra Singh has told you, and this will come back in tomorrow's my lecture, are that proteins have a number of co-translational modifications and post-translational modifications. There are more than 240 post-translation modifications. Some of them, like phosphorylation, have played a great role in the regulation of metabolism and a variety of cellular functions, including development, recognition, and other things. You have heard the detailed lecture from Dr. Yogendra Singh. Now, here and there, both in his first lecture as well as later, he mentioned about affinity chromatography, whereby some magnet-like things can be immobilized and then you can pick up a needle-like substance. Let us call the magnet antibody, or the pin that you wish to, steel pin that you wish to pick up can be an enzyme or any other thing. So now this leads to the general principles of affinity chromatography, and much more so on what is this, how do we make this immobilized enzyme or antibody to be useful in this technique. First of all, you have heard from Yogendra Singh the varieties of chromatography, which is a separation technique, very useful in biology, especially biochemistry. And one of the methods in this chromatography modifications is that you can have a single step purification, and that is known as affinity chromatography, because it exploits the great affinity between A and B. A can be something, B can be a magnet. So like that, exploiting that attraction or affinity, people have designed this affinity chromatography. During the lecture, somebody asked Dr. Yogendra Singh whether everything has to be in the solution. Yes, you can do it in solution also. I will show you in example. But otherwise, most of the time, the magnet is immobilized. That is, whatever you immobilize, antibody or anything, it is usually prepared as a water insoluble. Usually, the magnet is prepared as a second phase, as an immobilized form, solid phase. Therefore, this will be like heterogeneous catalysis or solid phase reactions. Now, however, when morning somebody asked whether things should be all in solution, most of the time, no, but we can also do that in solution. Here is an example. There is a hormone called luteinizing hormone, which is a protein, and this is found in the pituitaries of all vertebrates. I hope botany students understand this. Now this, how do we purify? There are great methods which involve months to purify this substance. But if we can do affinity chromatography, it can be done very fast. However, we made a modification of this. All of you are aware that when you do gel chromatography, you use a dye called blue dextran, which is a dextran molecule polymer to which a textile dye, Sibacron blue, is attached covalently. And people have used this blue dextran to determine the void volume in general chromatography, gel chromatography. What we did was, we made an observation that luteinizing hormone has a great affinity for this textile dye, Sibacron blue. And therefore, what we did, in a very unusual modification of affinity chromatography, we made this LH extract, pituitary extract, mix it with blue dextran solution. So both are in solution phase. And the luteinizing hormone binds to this. Now the whole thing, if you do a chromatography on a G100 or G200 column, everything comes out in the void volume. Because blue dextran comes out in the void volume, attached to that is luteinizing hormone. Normally, luteinizing hormone does not come in the void volume. It comes in the included volume. The understanding of this is from Yogendra Singh's lecture. And now what we did, we changed the pH, dissociated this, and again did chromatography on the same column. And now you see blue dextran colored blue, first peak, comes in the void volume, whereas luteinizing hormone comes in the included volume, separated from this, and it is very pure because it is a single monodisperse peak. You show this in orange color or whatever it is. And therefore, this is a clean purification method. Normally, we take 10-15 days to purify luteinizing hormone, whereas this method can be done in 1 hour. And therefore, this immobilized material, antibodies or enzymes or substrates or dyes or anything, it's a great technique. And therefore, let us spend some time in understanding how do we immobilize things on anything. First of all, immobilization means a substance present in solution is made into a water insoluble derivative. And therefore, it will become a solid phase. So it will become a suspension. So this conversion of a soluble phase to insoluble phase is also called as immobilization. Supposing we applied this to an enzyme, normally enzymes, proteinaceous enzymes are soluble in water, but we can, by some methods, which I will describe, you can make it a water insoluble derivative, or you can immobilize the enzyme. So this process of confining the enzyme molecule. Normally in solution, enzymes or any molecule keep moving within the solution. Whereas once you immobilize, it does not move. There is no freedom of movement. So this, to a distinct phase from the one wherein the substances and substrates and products are present, that process is called immobilization. How do we do that? Of course, there are a lot of benefits if you were to immobilize an enzyme. It protects from degradation, because normally if you take an enzyme, make a solution, then it will be subject to proteolytic cleavage. That's why enzymes become inactive. That is one of the reasons. But if you have it immobilized, this is prevented from degradation and deactivation. It retains, of course, the enzyme activity. You can repeatedly use it by recycling. It's very cost effective and it is greatly stable. When I say stable, thermal stability is conferred. And you can use as a controlled release agent also. The ability to stop the reaction rapidly by removing the enzyme from the reaction, you just filter it or centrifuge it, then the enzyme is separated from the product, because the products will be in the solution. Whereas enzyme is immobilized, therefore separation is very easy. How do we do this immobilization? There are a number of methods. We will see 4 or 5 of them. One, you can simply adsorb this enzyme or anything that you want immobilized onto a physical surface. The surface can be activated charcoal. It can be erythrocyte cell membrane, ghost. It can be anything. However, this adsorption, as it is called, is a physical process, and that's how the first method is physical adsorption onto an inert surface like charcoal or anything. In that, what was earlier in solution becomes adsorbed and therefore water insoluble. You can also do this on not an inert surface, but an ion exchange resin or an ionic surface. You can do so on nylon after partial hydrolysis. You can do so on DEA cellulose column, any ion exchange resin. Therefore, this is called as ionic bonding based adsorption. You can also link that by covalent bond. One of the problems with physical adsorption is there could be leaching by constant washing. And therefore, to avoid even that, one can covalently link this. I will show some examples of how to covalently link to a surface. Or there is a method by which we call it cross-linking, which means you can use a bifunctional reagent. I will give some examples and cross-link this enzyme solution or any protein solution, and they will get cross-linked and then become water insoluble. And the another method is you can put this enzyme, entrap it. There are ways of entrapping either in the lattice or as a microcapsule. All this we'll see more details now. Now the physical adsorption that I talked about, basically you sprinkle this solution on an inert surface. What are these inert surfaces? I mentioned about activated charcoal. And the interaction is basically hydrophobic force or sometimes salt bridges. Binding onto silica, clay, or ion exchange material, these are all weak interactions. And this is therefore called as physical adsorption. Of course, there are some disadvantages, limitations, because you can, one thing, of course, when you want to dissolve, it is very easy to do. That may be an advantage, but it is also becoming a disadvantage because when you wash with buffer, some of it will be leaching. So we have to be careful, but it is highly cost effective because when industrial scale production is, purification is attempted, people use this at a very large scale thing. In biotechnology, you would have read about some of these methods. However, this also can be done through ionic linkage, salt bridge. So if you take some water-insoluble solid support like DEA cellulose or carboxymethyl cellulose carrier, you can put this enzyme solution, because proteins are also charged molecules. If they are positively charged, you can absorb it on a negatively charged carrier. If they are negatively charged, you can absorb it on a positively charged carrier. However, both, whether you do it on an ionic surface or an inert surface, it is a pure adsorption procedure. So there is a possibility of leaking. Now, we can also link it by covalent bond. And here is an example. If you take any polysaccharide like starch or cellulose or sapodix or any one of these, they all are characterized by adjacent hydroxyl groups. In chemistry, these are called as vicinal hydroxyl groups. If you take such a material and react with a reagent called cyanogen bromide, which can be purchased, but can also be made in the lab if you know a little bit of chemistry, and that activates this resin. To this, if you add the solution of the enzyme or any protein, anything that has got an amino group, that will get covalently linked to this resin, and that is what is shown here as N-substituted carbamate. That is the kind of chemical coupling that you can do it. Two people you have to remember: Pedro Carthricasas, a Spanish biochemist, settled in America; Meyer Wilczek, another biochemist, but an Israeli fellow. These two developed many of these affinity methods using this cyanogen bromide activated sapodix or saffrose. Of course, there are advantages. The major advantage is that there is very little leakage. Covalent bonds are not easily broken. Therefore, tremendous amount of leakage is suppressed in this method compared to mere physical adsorption. Minor alterations in pH or ionic strength or anything will not create any problem in this. And therefore, as long as I know the chemistry of the inert, the so-called polymer support, we can activate stainless steel, nylon, cellulose, cotton, fiber, anything can be done. In fact, in large scale, we have covalently linked enzymes, antibodies, antigens, and all that to even nylon stockings or your shirt material or anything we can do. And mere dipping and removing, you can conduct a reaction. This is what is done in biotechnology, large scale immobilization and reaction thing. Now, this is, of course, morning in the lecture, somebody asked that these are all very expensive method. I think Dr. Haleem Khan asked, yes, they are very expensive, but I will give you a cheaper method later. We'll see what happens in biotechnology procedures where large scale purifications have to be done. Same principle is used, but the design is different. It can be made less expensive. But on a lab scale, small amounts, very pure materials can be gotten, and that is, of course, very expensive, and that's a disadvantage. There is another method of immobilization. This is a method using bifunctional reagents. Supposing I have a solution of an enzyme or an antibody or any protein, soluble protein, and then you add a reagent. What is this reagent? It has got functional groups, reactive groups. It can have 2 of them or more than 2 of them, and that reacts with 2 two similar functional groups in the protein, and thereby protein chains are cross-linked. When you cross-link protein chains, they are no more soluble because the size increases, aggregation, and therefore they become water insoluble. So you can filter them, wash them, pack into a column, you will get essentially immobilized enzyme, but by cross-linking. What kind of reagents you can use? I have mentioned here 3 of them, very popular: ethyl chloroformate, glutaraldehyde. Those of you who have done electron microscopy, remember this glutaraldehyde you can use for fixing. The principle is the same thing as we do in immobilization of enzyme. Tetranitromethane can also be used. There are many more reagents, but try to remember at least these 2 or 3, because if you are using it in future, you can use this. There are other methods also. I mentioned that in addition to physical adsorption, in addition to chemical linkage, coupling, in addition to cross-linking using bifunctional reagents, one can also prepare artificial cells. This is by entrapping the enzyme molecule or any such protein onto some sort of a capsular material. There are 2 ways of this. There are a lot of polymers which resemble a web, like you have seen for when you make tea or coffee, you filter it through a chalni. Exactly like that, if you take such 2-dimensional or 3-dimensional chalnis, you can get the enzyme entrapped in the lattice, and that gives rise to matrix entrapped immobilized enzyme or protein. On the other hand, you can also make this entrapment into spherical globular molecules like colloidal membrane or nylon membrane or artificial erythrocyte ghost. Anything can be done. So what you get is the enzyme in solution phase, but trapped inside a impermeable membrane, so that the enzyme stays inside. You can filter it, you can do anything you want. You just dip it in a substrate solution, you get products, but substrates and products are small molecular weight compounds. So they go in and come out, but your enzyme is separate. And therefore, this is a great method. A Canadian, but is a Chinese actually settled in China, Canada, TMS Chang, developed these methods. Therefore, we have different ways of immobilization. This I have already talked, lattice entrapment. And these make the enzymes, convert them from solution phase into a solid phase. That's why we call it phase separation. Another word to use is immobilization. I have compared all these things here. You don't have to go into detail. There are some advantages, some disadvantages with each one of them. The most important, of course, is the cost. So when you are working in a lab on a small scale, you can employ any of these methods. But if you are wanting to do an industrial scale production, purification, you can't use these methods like this. You need modifications. We will see this later. However, there are certain limitations, as I said already, cost of carriers and other things, selectivity problems, all these things will be there, but we will not go into detail. What you have to understand is only the principles of these methods. One of the things that people have done a lot of investigation, what happens to the enzyme compared to a solution phase when it is immobilized on a glass or on a nylon or a stainless steel or sand particles, siliceous particles, or any such thing. What happens to the enzyme? Is there any change in that? Yes, it becomes more thermally stable, and the kinetic properties also change. This is one such example. The kinetic Km value can change based on ionic strength, but that doesn't affect the enzymatic activity. The greatest advantage, of course, is its stability. So these things, therefore, you must investigate and then use this. Why do we have to do immobilization? I have already mentioned this is protection from degradation and deactivation. This is good for lab scale as well as industrial scale. All right. Now, leaching is something that you will have to always watch for. I will go through the same thing again because of some other points that I wish to tell. You don't see conformational alteration of the enzyme if you adsorb it. Adsorption is a very mild thing. It's very simple and very cheap. I mentioned about large scale production. This is the method for large scale production. People have taken huge cloth or some such thing on which it can be adsorbed, and you simply dip it in a vat containing the solution of the enzyme or anything. You will get this on a large scale. Few square meters of cloth can be used for immobilization. And that's why large scale conversion or catalysis can be demonstrated. All right. And you don't see in the physical adsorption method, you don't need any more chemicals. All you need is a surface and what is to be adsorbed on that. Okay. Covalent bonding you have seen. The limit is your knowledge of chemistry. You can link it to anything. You can link it to cellulose, textile cloth. You can link it to nylon, stainless steel. You name the polymer. I'll tell you the chemistry by which you can link. It all depends on your experience and your knowledge of chemistry. Okay. This is a figurative illustration to show you that a bifunctional reagent like glutaraldehyde, whose formula is or structure is shown above, and how it cross-links, because it will link aldehyde. Glutaraldehyde has two aldehyde groups. So it will link two amino groups of protein, one from one protein chain, another from another protein chain. So it will form a bridge, and therefore protein gets cross-linked. All right. Are there any other uses for these things? Yes. There are a lot of nanomaterials in which you can immobilize any protein, any drug, anything, and that is useful in drug delivery, which means for carrying these soluble things, but protected from degradation. So application of nanomaterials, novel supporting materials, these are made from inorganic material. It can be made from even organic material. You should know how to prepare a nano form of a given molecule, polymer, or simple inorganic compound. And if you do it properly, you can either adsorb on this, the drug, or you can include it as an inclusion compound. Both will be okay for delivery. That means for circulatory delivery. And therefore, this can be done on a large scale, although the size is so small, and biotechnology companies and communities have depended on this now. These are robust nanostructured forms, nanoparticles, nanofibers, nanotubes, nanoporous. Your Vice Chancellor was mentioning in the very first lecture in the morning that their department has done a lot of work on nanopores. And these are some of the examples which you can make it and use it as a carrier in the form of immobilized drug or enzyme or anything. These are tremendous applications. Graphene sheets can be done, nanocomposites, all sorts of things have been done now. The current status of versatile nanomaterial support for biofuel production employing immobilized cellulases and lipases is described in detail by many people. Nanomaterials will become an integral part of sustainable bioenergy production in future. Another application of these immobilized enzymes is in developing what we call as biosensors. You must have read it in a little bit of biotechnology. In the environment, in water, air, and everywhere, there are a lot of materials which are not good for our health, poisons, pesticides, and many other things. And these have to be measured. First of all, it has to be detected. You need very sensitive methods to detect. And these detecting mechanisms, if you do it in a particular way, using electronic circuit for signaling, you call them as biosensors. So an immobilized glucose oxidase can be used to detect glucose. Some of you senior people, if you are diabetic, you would have used a glucometer to measure amount of glucose in your blood sample or a blood drop. That essentially is a biosensor which has immobilized glucose oxidase, and moment glucose in the blood sample or anything comes near, it will oxidize it, and the generated hydrogen peroxide is made to give an electronic signal, electric current detected and therefore measured, and that essentially gives you, quantitatively you can do it, and this sensing is directly proportional to the amount of glucose within some range. Therefore, you can develop biosensor for anything on this earth, not just glucose, any metabolite, any pesticide, anything, any chemical. If you know the chemistry, if you know an approximate or a target or a sensing enzyme or an antibody or anything, you can immobilize that enzyme or antibody and then on an inert surface, but linked to electrochemical gradient generation, and that becomes a biosensor. And nowadays, these biosensors have also been miniaturized because now they are called as nanobiosensors, as I have explained here. These are illustrated here, any nanomaterial, but you should have a sensor first, and that is immobilized on this material, and that's how you get a nanobiosensor. But remember, the signal is always electrochemical. That means current measurement. Now, many a time, people who did basic investigation on this immobilized enzymes noticed that it has got a greater thermostability. Why do I say this? You need, in a country like ours, especially South India, you have heard a joke that South India does not have winter. It has 2 seasons, hot and hotter. Now, in primary health centers, all these detection methods are diagnostics. If they use these enzymes, basically it won't work because rural primary health centers don't have refrigerators, don't have anything. And the outside temperature is 42 degrees centigrade. Therefore, people use this in the form of an immobilized form, and that has shown thermostability. That means these strips are highly thermostable compared to solution enzyme. And this is what is important. I've given you some examples here. Pectate lyase from attenuated macrophomina is in the presence of hydroxyapatite nanoparticle. That means it is linked to that. Hydroxyapatite is similar to our dental material. Hydroxyapatite nanoparticles can not only act as chaperone, imparting thermostability, but can serve as a synthetic enhancer of activity of an isolated extracellular pectate lyase. We will not go into detail. All this is important in developing sensors as well as using enzymes on a large scale. And these are thermostable, as you see in the last line, 50 to 90 degrees centigrade, they are stable. Whereas a solution enzyme will not be stable like this. This is a table which compares all the things. I have shown this earlier also. Enzyme immobilization is one of the most promising approaches for exploiting enzyme-based processes in biotransformation. You must have heard in biotechnology that biotransformation is a big biotechnology industry where they exploit a unique enzyme which catalyzes a unique chemical reaction, but this enzyme is used as immobilized form. And so you have a continuous production of unusual products. One example I'll give you. Most of you know what is a steroid hormone. And if you look at the structure of a steroid, there is a 19th group, there is 11 group and 11 carbon and all. Hydroxyl group exists. It so happens that this hydroxyl group is in the beta form. Now, no organic chemist can do stereospecific hydroxylation, whereas these enzymes can do this very well. And therefore, people use immobilized enzymes, hydroxylases, to convert a non-hydroxylated derivative into a hydroxylated drug. It can be even a hormone. And that's the great advantage when you use immobilized enzyme versus organic synthesis. Okay. Now, this immobilization of enzymes can be extended to other proteins also. Because in affinity chromatography, you can use an immobilized ligand to purify the enzyme. You can use an immobilized antibody to purify an antigen. You can use an immobilized antigen to purify an antibody. Now, these antibodies are maybe polyclonal or monoclonal. Here is a diagram of how you generate monoclonal antibodies. You immobilize a mouse, special type called bulb C mouse, take out the spleen. I think all of you have some idea about animal internal anatomy. And this spleen is removed. And then if you macerate it, spleen cells come out. This is called splenocyte. And these splenocytes are producing antibodies. Now, if you take a cancerous cell derived from splenocytes or general blood myeloma cells, and then fuse these two, where did this fusion come into picture? Those of you who have studied cytogenetics, you must have heard that during the first decade of our 20th century, the cytogenetics people were asking a big question. If I take two cells and fuse them, which set of chromosomes will be active? It's a cytogenetics question, but they didn't know how to fuse cells. They serendipitously made an observation that virus particles can fuse cells. From that day, they were using Sendai virus, and then got the answer for their cytogenetic research question. However, we can now use chemicals like polyethylene glycol, and this is what Caesar Milstein did in 1975 to produce monoclonal antibodies. Here, what is shown is that mouse myeloma cells are fused with the splenocytes from the immunized mouse, and you get a lot of fused cells or individual unfused cells. By a nutrient selection medium, you can make sure that the hybrid cells survive, all other cells become dead. And therefore, dilute this and then clone the cell, which is hybrid cell. So it is called as hybridoma technique. So because it is derived from mouse myeloma, it is a hybrid cell derived from myeloma cell. So it is called hybridoma technique. Caesar Milstein got Nobel Prize for this. This is a picture of a monoclonal antibody. It has two valencies, but on the same epitope. And these monoclonal antibodies do not form precipitate with the antigen, unlike a polyclonal antibody. These details I will not tell you. Now, if this monoclonal antibody or a polyclonal antibody is immobilized, let us say on a solid surface, as it is shown here, if you immobilize an antibody, the Y-shaped molecule is antibody. Then if you have an antigen in two forms, one which is labeled, another which is not labeled, you can set up a competition between labeled and unlabeled, or you can add this antigen and then the antibody will bind, and then wash it and add another reagent called enzyme labeled second antibody that will bind, and then you react the enzyme and show the color reaction. You can get the label, or in this case, the enzyme is the label. You get the product and measure the color. This is known as solid phase immunoassays that we can design. You can call it also sandwich ELISA. We have done, many people have done a lot of work on these things. This is the sandwich ELISA where two antibodies will sandwich the antigen. What is the use of these immobilized antibodies or immobilized anything? These are, I said already, that if you make this with immobilized antibody, to sense anything, you call it an immunosensor. Otherwise, you can call it an enzyme sensor, anything. These are biosensors. Now, these immunosensors have tremendous biomedical application as well as analysis of environment. I don't know whether you have environmental chemistry, environmental biology, environmental physiology, or even environmental science. You would have been told that a lot of environmental components, toxins, pollutions, and other things can be sensed and quantitated by these immunosensors. You can use these as a cancer biomarker to measure them, cardiac abnormality biomarkers, because cardiac myoglobin, when it gets leached out, it means you had a heart attack. So people use an immunosensor to measure that. You can measure any metabolite in the abnormal metabolite in the blood or urine or in the environment, water bodies. You can measure pathogens, drugs. In sports medicine and sports and games, many of these sports people, to get a higher performance, they take drugs. Usually earlier it used to be growth hormone or testosterone, but there are a lot of narcotics also involved. All this earlier they used to do and escape and get rank or medal. Now they're all detected and they're banned from using these things. People have lost Olympic medal after finding out that they were taking these drugs for performance enhancement. People use this food inspectors to detect poisons in the food. Normally, if you buy a good goat meat, if the fellow is unscrupulous, he will mix it with dog meat. How do you know that he has mixed? There are safety norms, safety inspectors who can detect dog meat protein present in that thing and therefore can punish that shopkeeper. Environmental toxins are known, vitamins can be measured, lot of hormones, lot of antibiotics, anything can be measured by using immobilized antibodies, and therefore they're called as immunosensors. This also can be done by another technique. This is called surface plasmon detection method. All that is done is the antigen antibody complex is formed on a surface, and the thickness of that reflects a laser beam, light source, let us say, differently, and that can be quantitative signal to know how much is in the solution. And this is a physics people developed this, but it has become a very common method to measure or sense things. Now, therefore, we have gone through a particular aspect of proteins, which means how to immobilize proteins, enzyme or antibody or anything, it does not matter. If it is a small molecular weight compound, you have affinity chromatography developed through that. If it is a large molecular weight like enzymes or antibodies, you can also use this as immunoaffinity chromatography, but more important, they have been converted as biosensors, nanobiosensors, using many electrochemical methods for detection and quantitation. And therefore, this whole area of immobilized enzymes, antibodies, ligands is a major part of biotechnology for large-scale production. It is a major part of diagnostic methods for diagnosis. It is a major part of biosensors in environmental science to detect unwanted poisons, pesticides, and many other pollutants. Even gases can be detected in low quantities. It is also used in plant hormone detection in many cases. And therefore, this is a very, very, very useful method. I thought I will explain to you the principles and applications of these methods of so-called immobilization. That means converting a water-soluble compound like an enzyme or a protein into a water-insoluble. So you get a solid phase reaction. I have not touched solid phase radioimmunoassay, enzyme-linked immunoassays, which can be developed because time does not permit. So I think I will stop, but I want to field questions which they can ask. Thank you, Dr. Khan.
Yeah, it was a very detailed lecture on immobilization. Dear participants, if you are ready to pose the questions, you are welcome to pose the questions and I will read it out for sir. Right now, I have got one question, quick question. How this concept of immobilization has come into the thought, to say how this thought has come about the immobilization concept, where it got origin.
Yeah. Can you hear me?
Yeah, yes.
See, originally it started with in the field of enzymology, where biochemistry people, purify enzymes and assay them. But there was always a doubt when you take an enzyme and add the substrate and feed it continuously, you don't get enzyme reaction after some time. Most of you must have noticed it. Something happens to the enzyme. Jocularly, the biochemistry people say that enzyme gets tired after catalyzing a number of cycles of the reaction. But actually what happens is that the enzyme in solution phase undergoes denaturation. Therefore, people wanted to know how to stop this denaturation. And then the idea came that if you make it a water-insoluble derivative, which means immobilize them by all those methods which I have already described, this seems to be stable. And this inactivation of enzyme is also temperature dependent. That's why you notice that most of the biochemistry people have a refrigerator or a cold room in their laboratories, because they only believe that they can work at 4 degrees or 10 degrees. All right. Therefore, even in, for example, you do PCR. In the olden days, this PCR was done with a normal DNA polymerase isolated from organisms which are normal. Therefore, they are active only at 37 degrees. But in the PCR reaction, to dissociate the two strands, you will have to raise the temperature to above the melting point of the DNA curve or the hybrid molecule. And therefore, the enzyme is to get inactivated. Then they saw that this enzyme can be prepared from an enzyme, organism which is present in hot springs. You must have heard about hot springs. And one of them is the Thermus aquaticus organism. And the DNA polymerase purified from that is highly thermostable. And therefore, they got the idea that probably we can make artificially thermostable by immobilization instead of going for organisms which live in sulfur vents, sulfur wells and other things. And therefore, that is the source of this idea, how to stabilize. Some other time, we will arrange a lecture, how to do this artificially to get thermal stability, because in fermentation industry, you have to use a thermostable enzymes on a large scale. I am not doing this, but another workshop I am doing with Dr. Sindhura somewhere else near Hanamakonda. I think I'll have a speaker talk on this thermostability of enzymes and all, how to do this artificially using biochemical knowledge. That is the origin of this idea.
Actually, there is a question in the chat box. Advantages or applications of whole cell immobilization or enzyme immobilization.
Yeah. You see, in the immobilization method, technically you call it immobilization, but you see in the physical adsorption method, in covalent linking method, or in cross-linking method, you have literally immobilized it. But the fourth method, where I can take a colloidal membrane, which has got pores small enough that the enzyme cannot come out, but small enough or big enough for the substrate to go in and product to come out. Now what actually happening is, you can prepare this colloidal membrane in the form of a cell, that means spherical particle. Inside is all water or empty space. Now, during the preparation of this encapsulation, as it is called, if you have enzyme, then the enzyme will get trapped in the particle. Remember, we call it particle, but you cannot see them. They are in solution only, but this is not a actual true solution. It is a colloid. You must understand what is a colloid. So this is actually a suspension of these colloidal membrane spherical spheres, and the inside the sphere is the enzyme. It is almost, enzyme is freely floating there. And therefore, enzyme feels it is in its native state, so-called original conformation. So it is, although you call it immobilization, it is still in solution phase with all the advantages of immobilization. And therefore, using artificial cells is much, much, much better than other things. But this you can do only for macromolecules, because small molecules cannot be trapped like this. They will go out and come. But supposing in a bioreactor, I have, let us say, assembly line procedure where you have these immobilized, but in the form of a capsule present in a central reacting chamber. And then you have two tubes, one which is incoming and one which is going out. And you pass the substrate solution in a continuous stream. These will enter the bag, enzyme will react, the product will come out, it will come out of the another tube. Therefore, in a continuous bioreactor, you can use this kind of a method with tremendous advantage. That is the advantage of the so-called biocapsules or encapsulated enzymes. All this, if you read biotechnology, you will get a lot of application. Today, I am talking only on the principles, conceptual level.
Thank you very much, sir, for the answer. I had asked this question. Sir, to add on to that, to my earlier question, thank you for your insights, because the continuous bioreactor. But what I wanted to know was, for fermentation purposes, we can immobilize whole living cells as they are. For example, the yeast cells, or baker's yeast, for the purpose of fermentation, maybe alcohol or any other fermentation.
Absolutely.
So would that be, I mean, do you think it would be feasible and more economical than extracting enzymes from a cell and then using them for applications on a larger scale? That's
absolutely.
My question, sir.
You're right. But that kind of a cell immobilization is used not for the sake of an enzyme or something. It is for something else. For example, in artificial tissue making for grafts, people use now today we can have three-dimensional growth of cells in the form of artificial tissues. You can make a small liver, you can make a small pancreatic beta cells, islets of Langerhans, like that. In there, you have this immobilization of whole cells. It is not for the enzyme requirement, it is for some other purpose. And therefore, cells also can be immobilized in a variety of forms. Of course, you can always use them if in the form of an enzyme reactor, if you can again encapsulate the entire thing in, otherwise cells are mobile. So unless you restrict the mobility, you can't use it as a bioreactor. That is the disadvantage. But you're right, immobilized yeast can be done in large scale alcohol production. There are a lot of design changes. In the olden days, people used to use alcohol fermentation or alcohol production, ethanol production, using these yeast enzymes in a whole solution. Later, immobilized cellulose came, cellulase, whereby you can use materials like lignocellulose and other material, water insoluble. You can use them to generate monosaccharides, and from there, through fermentation, alcohol fermentation, you can produce ethanol. This is called bioethanol production. There are big industries on this, but please remember, when you use whole yeast cells and all that, you must know that external alcohol concentration will affect the yeast cell membrane. So beyond 4% concentration, you cannot produce more. That is therefore a disadvantage in using immobilized yeast cells or live cells. But if you use immobilized enzyme per se, alcohol production, alcohol concentration does not affect this. There is an advantage there. That is the comparison. Did I answer your question?
Sir. Yeah, definitely, sir. Thank you for the comparison. And I never thought that there could also be a disadvantage to this using whole cells like that. Thank you very much, sir. Thank you very much.
Sir, I have one more query.
Please.
Other than enzymes, are there any other commercial products which are made through this immobilization or entrapment?
Yeah, all your diagnostic methods, for example, you detect COVID virus through what is called as a rapid antigen test, RAT, right?
Yes.
I hope you have done the test on yourself.
Yes.
Kits are available.
Yes.
People, pregnant women, pregnancy can be detected by detecting HCG, a hormone in the urine of pregnant woman. And the method essentially uses immobilized antibody on a cellulose strip. And you have seen this thing, it is simply stick the strip or put a drop of urine on that, or in case of COVID, you put a drop of blood by pinprick, and then you see a color development or a line becomes visible. These are visual signal, and that is basically using an immobilized antibody. And therefore, you can use this immobilized antibody for anything you want. Detection of very functional proteins, especially hormones, or if sports people have taken enhancers like steroid hormones and others, you can detect very easily all these things. So it has tremendous application, as I said, in diagnostic thing. After all, medical practice has only 3 components: prophylactic use of vaccines, diagnostics, and therapeutics after the disease is detected. All these three things can be helped tremendously if you use immobilized enzymes, antibodies, antigens, ligands, all sorts of things you can do. Even dialysis can be done using immobilized urease. We have done that in our own lab. Although I am not a botanist, we purified urease from plant, then immobilized it and use it for dialysis of blood from patients, because urea is a very toxic molecule. Their own kidney is gone, and therefore we have shown there that you can immobilize urease. Urease is found only in plants, and this Cucurbitaceae family, we have done that, we have shown. Therefore, it has tremendous application. The more you see, the more you will read and enjoy. I only gave a glimpse of the range of this subject, which is very important in diagnostics, in biotechnology, and in basic biology.
Thank you, sir. Thank you for the answer. With your permission, I want to share some information on the protein's chronology. Can you read out? Sir?
Yeah.
So a book was published in the year 2001 by Oxford University Press, authored by Charles Stanford and Jacqueline Reynolds. They wrote a book on nature's robots, a history of proteins. A book was published of near about 150 pages. This book majorly covers the milestones in the story of proteins. So this book begins with the year 1789, where Antony Fourcroy was the person who was first to check out the albumins. That he said that albumins are of different types. And later on, it was the next step where the first amino acid was isolated from the protein in the year 1819. And the 20th amino acids, though we know that there are 20 amino acids, the first amino acid characterized among the 20 was leucine, and it took 1936, from 1819 to 1936, to discover the all 20 amino acids. The all 20 amino acids were discovered in a span of near about 100 years, near about more than 100 years or 100 years to discover the whole 20 amino acids one by one. And the first amino acid, as I told, is leucine, and the last 20th amino acid discovered or characterized was threonine. And one interesting point, what we noted is one of the amino acid named asparagine is origin, or its name has come from asparagus. We know very well asparagus. The roots of the asparagus were used to isolate this asparagine, and the name asparagus was made asparagine, as the origin was found in asparagus, and the name asparagine has come from asparagus. So in the 1809, asparagine was characterized from the roots of asparagus plant and made first time crystal form. And in 1837, it was a time when Gerrit Molder has compared the different protein compositions. Okay, Gerrit Molder has made the amino acid composition of different proteins for the first time in 1837. And in the 1838, it was the first time when the name protein was coined. For my students, I repeat again that 1838 was the time when the term protein, the word protein, was coined by Jacob Berzelius. And he, at that time in 1838, told that protein is a substance very important in the nutrition of animals. So Berzelius thought, or Berzelius has proposed that the protein is very essential in the animal nutrition. And he coined this term protein from a Greek word, proteos, which means that comes first or essential in the nutrition. And it was for the first time in 1840, the crystals of protein were made, like hemoglobin from earthworm blood smears. Hemoglobin from earthworm's blood was made into crystal, and then the plant proteins were first crystallized in the year 1855. And the molecular asymmetry and optical rotation was invented in the year 1874 by Van 't Hoff. Molecular asymmetry and the optical rotation was done in the year 1874. And it was a time in 1886 when the molecular weight of hemoglobin was chalked out. And in 1889, the first crystal protein from animal source, that is egg albumin, was crystallized by Franz Hofmeister. Later on, Hofmeister has shared Nobel Prize with Emil Fischer for discovering the peptide bond, or how the polymerization of amino acids take place, was cracked in the year 1902, and in the year 1905 or so, Emil Fischer and Hofmeister has shared Nobel Prize in chemistry. So in chemistry, the number of Nobel Prizes awarded are credited towards the protein structure, protein function, or protein, role of protein in various aspects. So in 1907 and 1908, there were international committees developed in the countries like United States of America and England to make the proper nomenclature and proper placing the protein in a correct part. The international committees were developed in the both countries, and till 1907 and 1908, the word proteid was in use. And these committees developed has replaced, or they have made the rule that it should not be called proteid. Instead of proteid, it should be called protein, was being fixed in that 1907 and 1908. And 1925 was the year where people have come to know about zwitterion and isoelectric pH by Adams et al. And it is again one more Nobel Prize which has been given to Swedberg in 1925, who used the segmentation velocity as the machinery of protein synthesis, ribosome. He was the pioneer who made the ribosome is involved in the protein synthesis machinery was done at that moment. And 1926, first enzyme was made crystal by J.B. Sumner, as just now sir told about jagbean, or it is also called as horse gram. In our own terms, we'll call it as a horse gram. And it was Astbury who has studied the wool from sheep and hair from other animals to crack the secondary structure of the protein. So for the first time, secondary structure of protein, in the morning session, we have seen that there are different structures of protein, like primary structure, secondary structure, tertiary structure, and quaternary structure. So Astbury was credited to know the secondary structure in 1930 for the first time in the wool from sheep and hair. And it was just the secondary structure involves the sulfur amino acid containing that cysteine bonds are present. And Bernal and Hodgkin were the people who did XRD, that is X-ray diffraction studies on the crystal form of pepsin. And then comes in the year 1937 when the electrophoretic methods were applied to serum proteins and found that the serum proteins are having some antibody type of properties, was coined or was investigated by Tiselius by using electrophoresis. So this was the first time in chemistry when the Nobel Prize was awarded to characterize or to find out the antibody characteristics in the serum proteins. And then Martin and Singe were the people who used chromatography as a tool for separation of amino acids. And again in 1952, Martin and Singe were awarded Nobel Prize. And then Moore and Stein have made the use of ninhydrin for the detection of amino acids in any mixture and characterize the amino acids detection tool or locating reagent. Ninhydrin was used in the first time in 1949. And Linus Pauling was a person who compared for the first time how a normal person's hemoglobin appears and how a sickle cell anemic person's hemoglobin appears. So this was done by using electrophoresis, and three-dimensional structure of protein was being made by Kendrew. And after this 1950s, we can see that the discovery of DNA was done, and people have started, after the proposal of central dogma, people have started doing a lot of molecular work supporting to the protein structures. Lippmann has got Nobel Prize in 1953 for working something related to protein synthesis, and Sanger also has got Nobel Prize for making the primary structure and composition of insulin, and George Pallady also was one who got Nobel Prize in 1974 for knowing the ribosome that are sites for protein synthesis and so on. So this was the information on protein chronology. So here comes our day 1 end. Over to sir.
Can I say something? Hello?
Yes, sir.
You see, what you have done is very good. We didn't cover all this because we cannot cover in one workshop everything about protein, number one. And remember, all that you have said is a historical information. And historical information, there is a lot of bias depending on who is writing the book. All right. Few corrections are necessary. Charles Stanford is a physical chemistry professor. He's the biggest authority on protein denaturation and renaturation, protein folding. Very great man. All right. But some of the things that he mentioned are wrong, completely wrong. The word protein was coined not by Berzelius, but by Mulder himself. All right. We didn't understand at that time. It is, of course, the literal meaning is it is the first, and therefore that name was given. You mentioned about Sanger. Sanger did the primary structure of insulin. That is the first time anybody did by using a reagent called fluorodinitrobenzene to detect and measure the first amino acid. The biggest question in biology at that time was, what is the holiness about sanctity about the position of an amino acid in a sequence? They thought it will change. Later, people found that the position of amino acid in a given protein is very holy, and there it will not change. What you see as a 12th amino acid will always remain as a 12th amino acid in a given protein. And to know that exactly, Sanger developed a sequencing method. You mentioned about electrophoresis. This Arne Tiselius that you mentioned developed what we call as free flow electrophoresis. What we all do today is not free flow electrophoresis, it is zonal electrophoresis. Arne Tiselius was a professor of chemistry in University of Uppsala. He had a great teacher called D. Swedberg. Swedberg developed ultracentrifuge and showed the sedimentation velocity measurements for macromolecules, colloidal solutions. He was a physical chemist and an engineer, and he got a Nobel Prize. His student, Arne Tiselius, developed free flow electrophoresis, and he got a Nobel Prize. Later, he became chairman of the Nobel Prize Selection Committee. His student, with whom I worked, he developed affinity chromatography by using that cyanogen bromide activated thing. Of course, he did not get Nobel Prize, but he is retired from professor of Uppsala biochemistry. You mentioned Martin and Singe and Moore and Stein. They are also developers of separation methods, which is a foundation on which biochemistry is based. Martin and Singe developed what we call as paper chromatography, and Moore and Stein developed ion exchange chromatography, and as you rightly pointed out, ninhydrin. When it comes to structure, when you talk sodium chloride or a simple molecule, there is only one structure. But when you talk a polymer like protein or nucleic acid like DNA and RNA, as Yogendra Singh pointed out in the morning, you have 4 levels of structure: primary, secondary, tertiary, and quaternary. When it comes to secondary structure, no doubt Linus Pauling contributed, but do not forget an Indian biophysicist, G.N. Ramachandran, who developed Ramachandran map, 5 angles of the peptide bond. I am giving a separate lecture somewhere else on this. Based on that, theoretically, he predicted alpha helix. Nobody is giving credit to him. He was nominated for Nobel Prize also. All right. Alexander Rich, another Nobel laureate, he was the one who spoiled Ramachandran's Nobel Prize. There are many stories in all these things. It is good to read history only to see inspiration, but not take it literally. There are many people. For example, you mentioned Astbury. He was the one who coined the term molecular biology, but nobody remembers him. He said powder X-ray crystallography of macromolecules is molecular biology. So molecular biology started as structural biology. Today we use regulation of gene expression as molecular biology. So the meanings have changed after the initial people coined this term. Incidentally, you mentioned Kendrew as the one who deciphered the 3-dimensional structure. It is not Kendrew, his colleague, senior colleague, Max Perutz. Max Perutz was a young boy of 19 years old. He was being talked down by his uncle, Felix Horowitz, in Germany. Felix Horowitz was a great protein chemist at that time, the biggest man in the world. And he was showing this young lad a small experimental under microscope. Under anaerobic conditions, he showed the crystals of deoxyhemoglobin and then said, look now, a magic I will do. He made, that means he breathed air on the crystal. Immediately the crystal broke. You never hear of crystals breaking with a whiff of air. And that is what instigated the young lad, the nephew, and he asked the uncle why it is breaking with just air. Then he said, I don't know the answer. You must go to England, work with a fellow called William Bragg and his son Lawrence Bragg. And this young Perutz went to England. He was 19 years old and asked these people that I want to do PhD. Why these people are great? They solved the structure of sodium chloride, which is only 2 atoms, and they got a Nobel Prize. And look at the gumption. They told Perutz, you solve the structure of hemoglobin, which has got 10,000 atoms. They themselves didn't know how to do this. It is Perutz who developed all the methods of X-ray crystallography. And then along, Kendrew joined for PhD. Both did. He allotted 1/4 of the problem, myoglobin, to Kendrew. He himself solved hemoglobin. Both got Nobel Prize. And Perutz for this got PhD. Can any one of you believe that he took so many years for PhD, but that also got him Nobel Prize? So all these are very interesting historical information, but one should use them carefully, whether there is accurately, correctly reported or not. Otherwise, we will be confusing students who will think that this information is more important than the growth of biology. In biology, the idea is important, not who did what at what time. All right. Kindly bear with me for these corrections. Thank you.
Thank you so much. Maybe the Oxford, I saw the Oxford Press book and I have collected the information. Thank you for making so many corrections. And listening to you is always encyclopedia, and it will be very interesting, and it will make your address makes us to feel very enthusiastic and listen so many things. Though they look like the past, but the people, how they have worked hard and how the things have come, the chain of developments, and how the sophistications in life have arises.
That is the most important.
Such lectures by you. I remember one of the lecture where you have told the concept of that hormone, where the two subjects were kept up, theft and that and all. I was very much, I had that video in my YouTube, and I always listen to that lecture, what you gave in our refresher course. So like this, if you keep on, spare some time with us, it will make us encourage us, and it will make us to learn so many things, what are there in past and what are there in future. So really it will be a walking encyclopedia along with us. So it is a good opportunity for us.
I will definitely do it. It is my delight. I am a delight, and I am a retired teacher. Anytime I will come. Only thing is, let the COVID pass. It has scared the shit out of everybody. So once this is over, I will come to your university. I will stay for a week or 10 days. You can ask me anything in biology. I don't like fragments of biology like botany, zoology, microbiology, or biochemistry, biophysics, genetics, physiology, and all. Everything we should know, but there is only one biology. That's how I introduced BSc honors in biology in Delhi University, integrated biology. When you read the course titles, papers, you wouldn't know whether it is botany or zoology or microbiology or biochemistry or anything. I don't use such words. Biology is important. Now, NEP 2020, Kasturirangan Committee has recommended that biology should be a single discipline. We should close all other departments. A lot of reforms are coming in the university system. Of course, central universities are doing it, but it is up to the state universities to change. We can't dictate terms here, because